Okay, good afternoon, everyone. Welcome to today's sustainability seminar. I recognize many of you here from our past seminars, so we're happy to have you here for the start of our fall seminar series. My name is Nancy Holm. I'm Assistant Director here at ISTC, and I organize the seminars along with Beth Muszewski. You probably see all the emails come from Beth. So the theme for this fall's seminars is contaminants in the environment. We have 4 seminars planned, and for those attending here, we have a list of those in the announcements out on the side table as you entered. And for those listening online, you can find those on our ISTC website. And the videotapes of the seminars are archived on our website also. Usually we try to have those up in a week or so after the presentations. We have over 8 years worth of seminars up there, so there's about 120 to 130 seminars on various sustainability topics. So with that, I wanted to ask everyone here to please silence your cell phones. And also, we'll hold all the questions for the speaker till the end of the presentation. Then I'll come around with the microphone, and you can ask your questions then. That way we have them recorded, and those online can hear the questions. And then for those viewing online, you can type your questions in at any time, and then we'll ask those at the end of the presentation also. So we're very pleased today to welcome our speaker, Ms. Dr. Diana Aga, who's the Henry M. Woodburn Chair Professor and Director of the Graduate Studies in Chemistry at the State University of New York at Buffalo. Dr. Aga received her Bachelor's of Science degree from the University of the Philippines at Los Banos and her PhD from the University of Kansas. Following that, she was a postdoctoral fellow at the Swiss Federal Institute of Environmental Science and Technology in Zurich, Switzerland. She began her career at SUNY Buffalo in 2002. Dr. Aga is also currently the editor of the Journal of Hazardous Materials. Dr. Aga leads a team at Buffalo that studies how various contaminants affect the environment, and her lab investigates techniques for removing antibiotics from wastewater, how plants, especially food crops, take up pharmaceuticals and engineered nanomaterials, and how levels of veterinary antibiotics in manure decrease over time through long-term storage or waste disposal processes like composting or anaerobic digestion. Dr. Aga also has extensive experience in analyzing persistent organic pollutants such as polybrominated flame retardants and how these compounds accumulate in the human body, Great Lakes fish, and the environment. So today she's going to discuss with us her research on emerging contaminants over the past 20 years. So please join me in welcoming Dr. Aga.
Thank you very much for the introduction and for the invitation. So I purposely scheduled this trip to be in somewhere like this time, September, because it's always very tricky to fly out of Buffalo between November to February when there's a lot of snow. But in the end, my flight was canceled yesterday, so I had to actually fly out of Buffalo to Indianapolis rather than here. And I ended up driving this morning, so it's still very challenging because of the rain. So I figured, because of the variety of background and from the audience, I figured I'll synthesize what we've learned over the past 20 years that I have been studying emerging contaminants since I finished my PhD. So that's been 20 years ago. So I was kind of looking back. I put a highlight of some of the things we learned, but mainly focusing on pharmaceuticals. So just a little background about my group, so in case there's some potential interest for collaborations in the future. I'm actually an analytical chemist, and but I apply it on environmental problems, and we study different contaminants from chlorinated, brominated compounds. I'll talk about antibiotics and pharmaceuticals today, including estrogens and hormones. And as maybe some of you have also seen that there's a lot of interest now on engineered nanomaterials being potentially ending up in the environment. So I categorize it as future pollutants, although there are now a few publications that actually show that there are now engineered nanomaterials getting into the environment. So our group is really interested in both fate effects and treatment, and to do all these studies, the first thing we do is to develop analytical techniques. And then we can study occurrence in the environment, including biotransformation and bioaccumulation in fish and wildlife. I'm also involved with a collaborator that does human health impacts. In fact, here I kind of put some of the publicities that we have received so far, and this is the work on PBDEs in milk, in paired samples, milk and serum from pregnant women. And then I'm also collaborating with several engineers looking at wastewater treatment plants. So my research is really highly collaborative and quite enjoyable to work with different backgrounds. So what are emerging contaminants? I think most of you work on emerging contaminants, but for those who are not, it's kind of a loose definition. It says that those are the environments that have been reported only in the last 20 years. But actually I have a slide here that I asked a student to look at. When was pharmaceuticals first detected in the environment? It actually dates back in 1970s, so it's interesting. But only in the past 20 years that interest have really increased, and that's mainly of the advances in analytical techniques. And they are causing adverse effects at very low levels, and they include both man-made and naturally occurring chemicals. So these are some of the list of emerging contaminants. Okay. So why are we interested in emerging contaminants? There's a wide range of impacts, and I just put here a couple that I think is probably the most important. One is the link to the emergence of antibiotic resistance. With all these antibiotics being introduced in the environment, this has become a major issue, and in fact there is an executive order in 2014, you can see here, about combating antibiotic resistance in the environment. So there's a lot of funding on this topic now. Because really at the low levels that we find them in the environment and constant exposure, that's perfect for making antibiotic resistance. The other one that I think most of you have also heard even many years back, so this is actually an old cover in the ASNT in 2003 where the first time they actually showed in the natural environment that at very low level, 5 to 6 nanograms per liter, you could really see an effect in fish when they are exposed to both natural and synthetic estrogens. So it's been studied many years ago in the lab, but this actually there is a Canadian lake where they introduced, where they exposed the fish in low levels of estrogens, and there was a collapse in the fish population. So after that, there's no doubt there's many other studies that came out showing estrogens in feminization of fish in the environment. So this was a timeline that I have asked my student to compile, and we published this in the Journal of Hazardous Materials Review just this year. You will see here that so these are the emerging contaminant research kind of highlights, and it mimics the development of instrumentation. So while my title is 20 years, 20 years of research, you could really see that estrogens was detected back in 1965 in the wastewater. And I don't know how they did it, but it's UV absorbance. For those of you who are involved in analysis, you might think that's really, I don't know, that's to detect estrogen with UV, it must be really, really high. So, but what's more interesting here is the commercial LCM-SMS, which is the liquid chromatography mass spectrometry that we are now using to detect many of these emerging contaminants. So back then, for the environmental chemistry analytical labs, we normally mainly use gas chromatography, so those are good for pesticides, chemicals that are not highly water soluble. But many of these pharmaceuticals are very, very polar, so they don't normally, you can't normally analyze them by GCMS, so that's why we didn't see them. But now with the LC-MS-MS being commercialized, more user-friendly, it gave us a lot of opportunities to actually look at pharmaceuticals in water. And so the first ever detected pharmaceutical is chlorfibric acid detected in drinking water back in 1994. But that didn't get a lot of attention. What really gave the scientific community much attention was this paper published by the U.S. Geological Survey in 2002, where they surveyed 126 streams, showing that the surface water is impacted by wastewater treatment plant discharges, and showing antibiotics, pharmaceuticals at low levels. And even then, there's still a lot of skepticism, and say, because the levels are low compared to pesticides, and really most of these pharmaceuticals are considered not toxic because, I mean, we do take them, they're medicine, so they're not like herbicide or pesticide that could affect, you know, insects or could cause cancer and things like that. So it took a while for it to really be of interest to the public and the government funding agencies. In 2005, for those of you who are involved in analysis, the commercial Orbitrap, it's a high-end, high-resolution mass spec, typically $600,000, so very specialized. It allows you to look at unknown compounds. So many of these compounds, it's a target analysis. You basically say, I want to look at compound A, B, C, D. Here, you don't know what you're looking for. You just kind of like non-target analyze your sample and say, I don't know what compounds are there. So it's very powerful. So now labs who have this are able to look at non-target unknown analysis. So this is now the kind of the future direction to see what else are there. So it's interesting also to look at the exponential increase in the publication. So we did a Google's Web of Science search looking at these various entries: pharmaceutical, effluent, wastewater, sediments, emerging contaminants, endocrine receptors. So we had help from our librarian. It's interesting how in over these 20 years we have now more than 3,500 publications, and I'm sure to date it's much more increased. And actually this didn't even include nanomaterials, so this is only pharmaceuticals. Okay, so what have we learned over the years? And disclaimer, most of these are from my group, so I did not include a lot of other groups. So, but there are a lot of other groups doing the same things, have found similar things, and here I will cite this group. Actually, I did Mike Thurman was my PhD advisor, so I worked with USGS during my PhD, and a lot of these are my colleagues. So I highlight. I'm not part of this. I just graduated back then, but this was the first time that we showed, or they showed, that human pharmaceuticals and personal care products are introduced in the environment because they are not removed during wastewater treatment. Okay. That's the first lesson. And so I just started my career as an independent researcher, and because of that, it kind of inspired me to look at this more in detail. And one of our very first publication is to look at the effect of hydraulic retention time and solid retention time on removing an antibiotic, tetracycline, which is one of the most widely used. And we found that solid retention time is actually very important, that the longer the retention time, the solid retention time, the more efficient it gets removed, most of the pharmaceuticals we're looking at. And really it's because in the higher sludge or older sludge, you proliferate the nitrifiers. So we kind of attributed also the high population of nitrifiers in the nitrifying activated sludge with longer retention time. And we have seen this, that so iopramide is an X-ray contrast agent that's used for MRI imaging, and it's very persistent, and it's supposed to be persistent because you're supposed to take it in and just excrete it 100%. So in the environment, it's also very persistent. So we use that as our target compound. Trimethoprim is an antibiotic. It's also very persistent, but we saw an enhanced biodegradation when you have high population of nitrifiers. And also this wastewater treatment It's in Amherst in Buffalo. It's kind of a two-stage wastewater. So this is kind of a special where after the conventional activated sludge, it goes to a nitrifying activated sludge. And we saw a significant increase in the removal. And then we went to the environment. These are some of the old work we have done. We basically look at several surface water receiving discharges. And we found that many of these surface water have different, just wide range of concentrations. And it depends on the wastewater treatment plants. I think now it's well known that the design is very important. But back then in 2006, everything was kind of new and unknown. So then we compared 4 different full-scale wastewater treatment plants, and we found that really the design and operation is critical in removing this. So therefore, it gave us some idea that maybe if we know a lot about which design and which operation is effective, maybe some of these wastewater treatment plants can be upgraded. Because, of course, we cannot just stop, taking pharmaceuticals, so we have to do something with the wastewater. And I work with 2 engineers that are well known on advanced oxidation and biological treatment. Carl Linden from Colorado State University, Colorado State University in Boulder, and Nancy Love from Michigan. We look at membrane bioreactors and advanced oxidation. So I'll show you a couple of these. So membrane bioreactors are also known. We've read literature, and in Europe, in big cities, they are moving towards the use of membrane bioreactor, and it's showing that it's more effective than conventional activated sludge. So we actually went to this pilot plant where we could split the effluent going to the conventional activated sludge and one going to the membrane bioreactor. And we measured what comes in and what comes out. And I'll just show you a couple of examples. Again, this is the trimethoprim that's very recalcitrant. So in the conventional activated sludge, about 29% only is removed. But when you go to membrane bioreactor, you can see here, this is the influent concentration, and this is the effluent concentration of trimethoprim. So in the conventional activated sludge, it's only 29% removed, whereas here it's more than 85% removed. And there are many compounds that behave this way. However, not everything is removed, and carbamazepine is one of the most persistent pharmaceutical. It's an anti-epileptic drug. So you can see here what goes in is the same thing as what comes out in both conventional and membrane bioreactor. So, okay, so we know that MBR is good, but it's not perfect for all compounds. So what else can we do? So this is where we work on advanced oxidation process. So this is actually a municipal wastewater in somewhere. It's outside Michigan, where they have a UV oxidation. Now, UV oxidation doesn't remove pharmaceuticals unless you apply hydrogen peroxide. So the hydrogen peroxide generates hydroxyl radicals from water, which are very reactive, and they react with the pharmaceuticals and convert them to more biodegradable compounds. So that's what we found from this work. So in fact, we did it also in the lab using a radioactive. So this is a carbon labeled, a C14 labeled carbon, because we wanted to know what's going on. We wanted to follow the degradation products. So we mixed radioactive carbamazepine with high amount, and we did this. So you might say that's really high. We did this mostly for analytical purpose, because we wanted to analyze the biodegradation products. So we compared something where we have advanced oxidation and without advanced oxidation, and then after advanced oxidation, we subjected them to biodegradation in activated sludge. Okay. We found out. So I'm going to go through these graphs here. So this is the control, where we just look at the typical biodegradation of carbamazepine, and we measured the radioactivity. You can see here we observe from to day 25, there was no change. So there's no change in the radioactivity in the liquid. So there's no biodegradation, and we expected that. That's what we see. Now, if you first subject it to advanced oxidation with UV and hydrogen peroxide, this red portion here, these are CO2. So some of them are being mineralized to CO2 and degraded when you subject them to biodegradation. So we monitored up to 25 days. So there is a potential promising technology. However, the disadvantage of this is it's highly energy consuming. So there's still a lot of things that needs to be done to make this useful or practical. Okay. This particular figure here shows you these peaks are the parent carbamazepine. It basically shows you here that from day 0, day 9 to day 25, there is no change. The area is the same, so it's not degrading. What changes, these new peaks here appeared when we did the advanced oxidation, and you can see that these metabolites or degradation products are the ones that are removed. So in summary, what I'm showing you here, so this is the parent compound that is typically not biodegraded. During advanced oxidation, it's being converted to these products, which are now biodegradable. So that's how it becomes CO2. Now, the question is, we did this in the lab. Can you really translate it in the wastewater treatment plant? So we did this in Colorado, where we put a pilot plant that received, so this is kind of like a schematic diagram. They used, so I didn't do this, but the engineers there that we were working with was low pressure and medium pressure, and they took the secondary effluent, subjected this to the advanced oxidation, and we analyzed the samples. And this is the real picture, basically. So we have the UV lamp and then the biofiltration. We also compared it with nitrate, because nitrate actually also produces hydroxyl radicals, the same thing as H2O2. So there's several things we did this. But what I really want to show you here is it's not only working for carbamazepine, but we look at other compounds, and it also works for other compounds. So you could kind of generalize. So this is iopramide, the other compound that is very recalcitrant that I mentioned earlier. We also use a radioactive compound, and again, we use that to help us identify byproducts. So we did the same experiment I described earlier, and we saw the same thing, that if you don't do advanced oxidation first, and we monitored actually up to 42 days this time, there was no degradation at all. And then you do the same thing with the UV, and here the red portion is the CO2 that's mineralized. Okay? And again, using LC-MS, so we published this more recently, we could identify all the byproducts. This is the parent iopramide that is not degradable. During advanced oxidation, we've identified all these different transformation products that are biodegradable. So this is a promising technology that can be, we haven't done more on this. We tried to actually, we tried to make a proposal to NSF, but I think the reviewers were thinking that it's very energy intensive, so we have to think about how to make it less energy intensive. And that's the engineering part I don't know so much about. But I think it's promising, and we're going to try to do this in the future. So lesson number 3, veterinary drugs are also introduced in the environment from non-application of manure. I think all of you know that. And I was mostly interested with the dairy manure, and that's because New York is the third, actually now it's the 4th largest. Idaho did it, but we have a lot of dairy farms in New York. And I got this from the EPA's website. You can, I think, again, most of you know, they produce a lot of manure per cow. So this is, for me as a chemist, I was also very surprised. And then when I give this talk to a bunch of chemists, they're all always impressed how much manure dairy cows produce. So I think most of you are not surprised by that. But we also use fertilizer manure to fertilize the field. It's a good low-cost fertilizer and improve soil quality and reduce erosion. So it's a good practice. However, the manure is contaminated with antibiotics that are used in animal production. And this is a relatively new data. This is only sales, so it doesn't really tell you what kind of use they are. But this is the antimicrobials sold for food-producing animals. And you can see that tetracyclines is the most used, and so we have a lot of interest in analyzing tetracyclines. Ionophores are the second one, but there's less interest in that, and that's because it's not used in humans. So I think a lot of people are convinced that it doesn't contribute to the production of antimicrobial resistance, even though it's widely used. So so we are also looking at penicillins and macrolides and sulfonamides. Admittedly, we don't analyze aminoglycosides. It's very, very tricky to analyze. We still haven't figured out how to do that, and so on. So it didn't show up here, but this was an old paper that we analyzed actually when I was still working in Nebraska. This is a field plot applied with manure from swine, and what we learned from that study back in 2002 is that tetracyclines, sorry, it didn't show here, these are tetracyclines. So tetracyclines are also very tricky to analyze, and even now with the LCMS, it's very laborious. So we tried to use ELISA, which is an immunoassay kit that you could buy commercially. It's much easier, sample prep is easy. But then we saw we were going to use it for screening. We saw that everything has high levels of tetracyclines, and it actually persisted for more than a year. Then we analyzed it by LCMS method, which is more laborious. We saw that they don't agree with each other, and levels for LCMS were actually much lower compared to ELISA. So we tried to look at why that is the case. We found that tetracyclines are actually transformed to many different isomers, but it's still tetracycline, and some of them are actually more biologically active than the parent compound. But because the immunoassay, the ELISA, is class selective, it picks up all kinds of tetracyclines, so that's why the levels are high. Whereas the LCMS is more specific, it's more targeted, and we only analyze for 3 tetracyclines. So that's why there's this big discrepancy. But what we learned from this is that tetracyclines are persistent in soil. So what can we do? People are interested in removing antibiotics because manure is very important to fertilize the fields. So we're thinking that maybe the advanced treatment systems like anaerobic digestions would remove it. But we learned actually that even the advanced manure treatment systems, they do not remove. So we look at antibiotics, hormones, and antibiotic resistance genes. And this is a facility in Western New York that has 2,200 cows. And this is actually a kind of a unique anaerobic digestion because it has a pasteurization process. So we were thinking that maybe during the pasteurization it will degrade the antibiotics, but it didn't. So we collected from the manure, raw manure, and then after pasteurization, and then after 22-day anaerobic digestion. We analyzed for all this, but we only detected tetracyclines and sulfonamides. So we look at all the different sulfonamides and all the different tetracyclines. We also look at macrolides and so on. But I'll show you the data we found. So this is actually, again, a surprising observation because we were expecting it to decrease. But you can see here, this is the raw manure, and this is chlortetracycline, epichlortetracycline, tetracycline, and epitetracycline, the transformation products. So the concentration in raw manure is actually smaller compared to the after digestion. This is not significantly different between pasteurizations. There's no change during pasteurization. But after digestion, we have this increase. Like what happened here? we discussed it with some people that are familiar with the digestion and so on. I think the hypothesis that we came up with, and it's reasonable, during post-digestion you break down all these solids and it releases the tetracyclines, because tetracyclines are highly sorbed on particulate matter. So you kind of digest it and disintegrate it into smaller particles, and you release in the liquid, because we only analyze the liquid, so we centrifuge the manure, we analyze the liquid. And so it released the tetracycline. But so again, the bottom line is it didn't really remove the tetracycline, this is what they apply in the field. We've also sent our samples to Amy Pruden from Virginia Tech, who does microbiology, and she looked at different tetracycline or different resistance genes here. And we also saw there is no change in the resistance genes between raw manure, pasteurization, and after digestion. So that wasn't what we expected, but that's what we observed. There's another system in New York that is newly installed. It's called the Livestock Water Recycling Unit, and we just started this work. It's funded by the USDA. So I will give you some data that are unpublished. They sell it, or they in their website and all their publicity, they claim that you can turn manure into a drinkable water. I met this guy. He said, I can drink it. So he took it. I said, how do you know it's clean? It's clear. I said, okay, but you smell it. It still smells manure. I said, have you analyzed it? No. I said, okay, so how do you know it's clean? And we actually talked. So this is by a Canadian company. Maybe some of you may know about this. They said they've now installed 7 of these in the U.S. And actually, this is the first one in New York, and they're installing in Nebraska, I think. And when I talked to the owner, they said they put a lot of money, more than $1 million, and it's only for public perception, because they don't want to upset the people around them. It removes the odor and so on. So they're willing to put money in this. So I was just curious. So we collected samples from the different parts, because it's kind of worrisome for me to know that they're drinking. Actually, I talked to the engineer from Canada when I called them. He agreed to talk to me. I asked him, would you ever drink that? He said, no. So I said, how do you know it's drinkable? Well, the engineers that install them, they kind of, and the owner actually also drink it. So, okay, so we'll see. It's a black box. I don't know a lot about what's going on there. It's all proprietary. Admittedly, I couldn't know what is this. So they have a liquid-solid separation, and then the liquid goes to this, they call it a phosphorus enrichment, and they put a polymer that's proprietary, and the product is really gooey, and they said it's biodegradable, so they put it in the environment. And then they have this nitrogen recovery unit. I also don't know what goes on there. And then they have a reverse osmosis. The membrane is also proprietary. So we collect here in the final effluent, but they don't really use it for drinking water. They put it back to the storage lagoon, and they use it to flush the barn. So it just gets recycled. And so we have all these different. So we analyzed for ionophore. We're not done with the antibiotic analysis, but we're done with the ionophore analysis. So this is what I could show you. This June 15 is before installation, and so we were able to collect a few months after installation, and then a year after. So you have 4 sampling points here. For monensin, it is, I guess we'll just focus on something. So the flush lagoon where they collected, there's not so much change in the concentration, but freshly separated solid, so you could see there's not so much change in monensin really. I think it's a kind of a ballpark. But in the liquid part, so we separate the manure, solid and liquid, we separate. There is maybe a slight change here now, but it might take a long time to do this. Also, we don't have any record of how often they change the membrane, so I think we're going to go back to the farm owner. The good thing is the farm owner is very interested in this and so willingly just have my students come and so on. So we're just now collecting data, and then we're going to go back with them and see the records when they change the membrane and all that. And then as I mentioned, I don't have the data for antibiotics yet. So, but I think this one particularly, so this is the stored separated liquid. There is a big decrease in that area, so maybe it will be over time. So I'm very curious about that. Okay, lesson number 5. If you are only targeting for a compound like hormones, for example, like 17-alpha estradiol, which is the main hormone in the cow manure, if you're only targeting for those and they decrease, it doesn't necessarily mean that it got removed. It just means it's transformed, and that's why we have to be careful about the word removal. So here's why. So the alpha E2 is the main, in the raw manure is the main estrogen, okay? And if you look at it in the raw manure after pasteurization and after digestion, you can see that it's being removed because of the decrease in the alpha. But then if you analyze for the metabolite, estrone, it actually increased. Okay, so it was started from really low to really high. So it's just being converted. And we looked at the estrogenicity actually using yeast estrogen assay. It actually increased during post-digestion. So the estrogenicity of the manure was lower in the raw manure and higher in the post-digestion. And the reason for that is in the raw manure, you have the E2 alpha as the major compound, and it has lower estrogenicity than E1. So this is converted, there's all this interconversion. In the post-digestion, the main component is E1, and it has a higher estrogenicity, okay? Now, different animals have different proportion of this. So for example, in humans, we have the E2 beta, and that's actually the most estrogenic. That's why in human wastewater treatment plants, the estrogenicity is much higher because of this is higher in the human hormones. Okay, so it's very important to look at transformation products, and we have also observed this in other work in the human wastewater treatment plant, where we like using radioactive compounds. We actually use radioactive compound to also monitor what the transformation products are. This is ethylene estradiol. It's the active ingredient of birth control pills, so it's a synthetic estrogen, and it's very estrogenic. We saw in this pure culture, so these are in the lab, that over time you can actually degrade or you can remove EE2. So this is day 0 to day 19, and it's gone. But then you form this compound, which is still has the ring responsible for estrogenicity. So you're just converting it to another compound. It has a lower estrogenicity, but it's still persistent. And in fact, what you will see here is in day 19, it's much higher in concentration. Okay. Again, I mentioned we like looking at radioactive chemicals. It helps us identify the transformation product. So this is a radioactive chromatogram. Without this, the LCMS will not be able to detect it. So it shows us these new peaks forming. So EE2 is the active compound in the pill, and when you degrade it with microorganisms, you will see new peaks forming, and you can identify it by mass spec. So that's how we like working with engineers, working with chemists, because we can identify things that are not very easily identifiable. So we wrote two kind of critical review about highlighting the importance of metabolites and also trying to redefine the word removal, because removal doesn't mean complete disappearance. So lesson number 6, emerging contaminants are spread in the environment and may potentially reach the food products. So part of our USDA project is to look at antibiotics going to the food systems. So we know that wastewater treatment plants contribute also during water use, using irrigation, also manure application. It can be taken up by plants. Okay, there are several literature now showing that in the greenhouse, hydroponic, so now we want to make it in the field. It's always better to actually demonstrate it in actual environment. So we collaborate with this, and this is actually a different kind of project. We are working with a nonprofit organization called Rich Earth Institute in Vermont, where it's really interesting. These are just local neighborhood collecting urine and using this to fertilize their fields. And the idea is to, instead of source separation, basically, instead of putting all this urine into the wastewater, which then you spend a lot of money and energy trying to remove nitrogen, why not isolate the urine and use it directly to fertilize the field? Of course, there's this gross impact in human thinking, and that's actually, we just got funding from NSF on, it's called under the program Infuse, which is Food, Energy, Water Nexus. And that has a social behavior aspect, huge aspect, to kind of convince people to adopt this practice, I guess. So they help us in this field. And the question is, if you apply urine as a fertilizer without any treatment, what happens to these pharmaceuticals? Are they taken up by plants? So we just use a whole bunch of different pharmaceuticals with different behaviors and different persistence and different uses. So it's a wide range, ranging from antibiotic to lipid inhibitor to these are psychoactive drugs, pain relievers, and so on. Okay, so again, different chemistry. And again, the idea is to separate urine so that you don't put it in a wastewater treatment plant, so you can already use it as direct fertilizer. But again, it's probably not a viable tool large scale, so we are working with a wastewater treatment plant that actually does struvite formation. They precipitate this the nitrogen and make it into solid fertilizer, and they actually sell this already as a commercial product. So we wanted to see what the difference between direct application and precipitation with struvite. Okay. And part of the study is to look at actual food that was grown in these plots that were fertilized with either struvite or urine. And we harvest them just like we normally do as a market ready. And we use lettuce as one of our above ground and carrot as below ground crops. So I'll show you some of our results. So in the urine, of course, we needed to analyze what's in the urine. And expectedly, we have much higher pharmaceuticals in urine than when you analyze the struvite, the precipitated fertilizer, so significantly lower. Okay. These are acetaminophen, caffeine, always high. Erythromycin, carbamazepine are mostly detected because they're very persistent. And then you have this over-the-counter drug, naproxen, diclofenac, ibuprofen. Okay. So what happens with when you compare lettuce planted in struvite fertilized versus urine fertilized, again, we kind of expected this. It was higher in the lettuce, okay, so that are planted in the urine fertilized, but it's, if you look at it, it's really nanograms per gram. It's very low, nanograms per grams per dry weight. But as you can see here, the struvite treated is again much lower. In the lettuce, we even tried to see, some people like their carrots peeled, some people like their carrots with a peel or just the core. So we again tried to analyze the core and the peel, the carrots. Let's see. For urine, again, the highest one that we see is acetaminophen. And again, these are really low concentrations. And there's no difference between the core and the peel. That was something that we learned. So it just kind of distributes itself to the core of the carrot. And again, the struvite is much better in terms of. So what does it really mean? So I think some of the people that we're working with, they still want to see that the urine can be directly applied, it's practical, it's on a small scale, it's usable. So they ask us, can you calculate what it really means that we can understand? So I asked my student to do a back of the envelope calculation on the most, using the highest concentration of acetaminophen and calculate it in terms of how many carrots you need to eat to take a 500 milligram acetaminophen. And you must consume 150,000 carrots to be equivalent to 1 pill. So in the big picture, it is negligible, you can argue. Okay, so lesson number 7. A lot of this couldn't be done if you don't have the analytical chemistry. So there's a lot of challenges, and that's why it took us a long time to know more about this. There's a lot of challenges in analyzing emerging contaminants. The first one actually is finding a student who would like to collect manure samples. Always difficult to attract them to say, we're going to go to the field and collect manure. The second one is sample preparation. You don't just really collect manure and inject it in the instruments. That's the hardest part actually. So it's easier to find students than to solve the analytical problems, especially, and even in the winter. This is one of our students who are looking at this. In fact, our very first publication is just basically addressing all the challenges in tetracyclines. It's very difficult to analyze, as some of you may know. First is because you can't really have a good recovery. In fact, you kind of just have to settle what is good enough. So we tried many different pH, many different temperature, and you can see here you almost just have 50%, below 50% recovery. So you just have to, EPA actually has a separate method only for tetracyclines. So that's one challenge. We clean it up with this technique. For those of you who are not analytical chemists, we use solid phase extraction, and these are very expensive cartridges, but you use 2 of them, tandem, different packing material. And what it is ideally you would like to have a clean water or clean sample to inject in the instrument, but this is normally what you get without a lot of cleanup. So in the end, you still, so just to kind of give you an overview, with all this different procedure, your cleanest sample is still yellow for a manure. And that is always difficult for the instrument. You clean up the instrument, your instrument break, So very expensive research. And why did we not see pharmaceuticals back in the years before LCMS? Well, before the LC, the liquid chromatography, has a UV detector, so they only look at the absorbance. If you inject this in the UV detector, this is what you see, this big blob, and you don't see anything. But with the mass spec, you could actually see every peak corresponding to the tetracyclines. So, so now almost every environmental lab has all these expensive instrumentations, and even the 2002 paper by Colpin et al. From USGS, they only use single quadrupole. That's the cheapest one. For now, I think hardly anybody uses single quadrupole because they actually saw a lot of errors in that paper. There were some companies that kind of criticized that their concentrations were very high in the surface water, that if they calculate back to the sales, it doesn't match. So there's a lot of, that's why it's actually highly cited, but some of the citations are because they're being criticized of the method. So if you want to be really accurate, I think you don't want to use single quadrupole in this messy research where you're dealing with manure and wastewater. Ion trap is also good. There's advantages, but the detection limit is not so great. So triple quad is kind of the typical LCMS. Nowadays, people are buying Orbitrap. It's, if you have $700,000 or to $1 million, that's the way to go. This FTICR, this is the instrument. It's great for low-level detection, but it's good for identification of unknowns. As I mentioned, ion trap is also good, but our very old publication in 2005 used ion trap. But now we are using triple quad because the detection limits are much better. In fact, before we would have like 71 detection limit in nanograms per liter. Now we could analyze 2 nanograms per liter. And this is even an older method. You could even go better if you have the newer instrument. One thing good about ion trap, or if you have the capability of doing MSMS, so this is like you isolate the chemical and then you fragment it, is that it avoids false positive. And for those of you who are not chemists, I will try to kind of like walk you through this. And this is just a fingerprint of the element or the molecule. So this is erythromycin spiked in distilled water. So we know it's there. You know this is the peak, and you know it comes out at 24.6 minutes. When you do the MSMS, it gives you these fragment ions. And this is a signature ion of that compound. So you could say, if erythromycin is there, you will see 522 and 558. If it's not there, it shouldn't be called erythromycin. When you analyze wastewater effluent, it's very messy. So you could see this peak, and if you didn't have MSMS, and you were only relying on the retention time, you might identify it wrongly and say, oh, I have detection of erythromycin here because I have a peak in 24.6. Now, if you do MSMS on that, and this is our actual data, we didn't see these 2 peaks. So if I didn't have the MSMS capability, I would have identified this as positive and have reported a wrong erythromycin. And this is something that had been criticized on some of the older papers where they only have single quad and they didn't have MSMS. So it's very important that the analytical chemistry is carefully done. The other thing is quantification. Again, some of the older, very, very old, I say the 2002 papers, they didn't have a lot of the so-called labeled compounds, deuterated compounds. So when you quantify them, the matrix actually affects quantification. So surface water, drinking water, groundwater, if you spike them with the same amount, they will all have different signal quantification. And I illustrate here. So this is a 100 parts per billion spiked water, standard here. And so the height is proportional to the quantity. And this is the 150 B in water. If you spike the same amount in soil extract, the signal is significantly suppressed. So if you weren't careful, you will underestimate the quantity of your compound. So we do this so-called isotope dilution, where we buy standards that are labeled with deuterium or C13, and we spike it from the beginning and go through the process. So everything is quantified accurately. The disadvantage of this is that these labeled compounds are very expensive. So 10 milligram is $500. So that's the disadvantage. But you can be very sure. I'm going to fast forward here quickly. I see that the time is running out. So lesson number 8, no one size fits all method. What I mean here is that we thought we have a really good method for identifying estrogens conjugate in tetracyclines in soil and water. We published this. We even use it for milk and all that. So we got contacted by a person from Maryland to analyze poultry litter. I said, yeah, we can do that. Sure enough, we couldn't. This doesn't work on poultry litter. So we need to revalidate everything again. And it turned out we can't do it without derivatization because the matrix is so dirty and the levels are so high. So we ended up going through this elaborate derivatization to do the method. So don't assume that your method works for one thing and it can be used for others. I'll skip that. Lesson number 9, so I'm almost there. I have only lesson 10. That sometimes the obvious is not the best approach. And this is mostly for the analytical chemists here. This is so-called wrong way around ionization. What it is there's a positive ionization and there's a negative ionization in mass spec. And typically, a lot of these are in the positive ionization. And to make them positive, you want to make it in an acidic pH. That's the obvious thing. But actually, we found that if you make it basic, the signal is higher. So it's the wrong way around ionization, which is not expected. Assume that the typical thinking is always correct. In fact, another thing is if you have a low detection, if you want to detect low levels, again, the tendency, the obvious thing is, oh, I want to detect lower concentration, so I should extract more manure. That's right. If you extract more, you should be able to detect at a lower concentration. It actually works against you because of the matrix. So I'm just going to forward this. So if you have the 1 gram, which is you expect it to have high signal because it's 1 gram, it actually has the lowest signal because you extract a lot more dirt. Whereas the lowest manure actually has the best signal. So it's the opposite of what you're expecting. Okay? And then I emphasize again the importance of transformation products. So again, if you go to the farmer, they say we apply tetracycline, oxytetracycline, chlortetracycline. So if you only measure these 3, you are missing a lot of the transformation products. So 1 tetracycline molecule, actually after you put them in the cow and they transfer in the environment, you have at least 3 different species of this molecule. So you have to look for this as well. And interestingly, the dehydration product, so this is just loss of water, is actually more toxic than the parent compound. So again, that's the unobvious thing. And then finally, I don't know what is obvious, but we have to figure out, like this is one of our farms that we're working on collecting. What do you really want to collect sample? We don't know yet. So and here you have this wide lagoon. We collect sample here. It doesn't mean it represents everything. So it's a lot of challenges. We could be criticized really like, how do you know that what your sample here is the same as here and there? I mean, do you really need to sample everything, or do you want to sample only the ones that is being used in the field or the runoff and so on? So we're still trying to figure out all this, what's important. The final lesson is most important. I couldn't do all this without good students and without the funding that we received from various funding sources, USDA, EPA, NSF, Water Environment Foundation, and then the New York State Pollution Prevention. But all these are my current students, but I have many more that are now working for many of these government agencies. And actually all those are their work, not mine. So that's all. I thank you all for your attention. I'd be happy to answer any question. Oh, okay. Sure.
I take it from your example of occurrence that there haven't been studies of showing the adverse health impacts on humans from what your analytic chemistry is able to detect water or foods.
Correct. So the question is if there's any human studies done from the pharmaceuticals detected in food crops. And correct, there has been no study. It's just been assumed that the levels are so low. And so there's not a lot of interest yet to this end.
Advanced oxidation technology, we're talking about the release of CO2.
Mm-hmm.
It sounds bad in terms of a greenhouse gas. What, how much of an increment on overall CO2 or methane releases from the treatment plants?
Yeah, I think it's just very, very small because these are micropollutants. So when they are mineralized CO2, these are extremely low, and I don't think they should be a concern for greenhouse gas formation. Yeah.
Okay, thank you very much. John?
I'm just wondering if there have been any studies of, let's just say, the sewage coming out of demographic locations, such as, do communities with a whole lot of young people have a different wastewater composition than, say, a geriatric place in Florida? And if so, is it possible to say, take an old folks home in Florida and essentially pre-treat the sewage before it enters the main sewer line?
Yeah, that's a very good question. In fact, it is now being done. There's this group of other researchers, they title them their work as sewage epidemiology or sewage forensics. And most of them are looking for illicit drugs in wastewaters to see where the population with a lot of drug use. Some also are doing exactly what you suggested, actually, to maybe have a local treatment for hospitals so that they don't get mixed with a larger population. Yeah, to make it more, efficient. So exactly, yeah.
Do you know where some of those studies with the
So the forensic one looking at illicit drugs, the person that does a lot of that is Jennifer Field from Oregon State, but mostly the work is done in Europe, in Amsterdam, places like that. But there's a couple of groups here. I could think of Jennifer Field as one of the pioneers in that in the US. I can't think off my head about this hospital because I think there are many other groups that are actually looking into that kind of approach. Yeah.
Okay, other questions here? Spin out, come around with the microphone.
Thank you for your presentation. I was wondering, with your analysis of micropollutants in raw manure versus some of the post-digested solids, you mentioned that in some of the samples after anaerobic digestion they actually increased in concentrations. Did those analyses account for sort of the volume concentration factor associated with, say, anaerobic digestion, where a lot of the solids would probably go
No, we didn't correct it for that. That was actually something that we looked at, but we freeze dry them also. They're based on dry weight. Yeah. Yeah.
So you sort of made the point that, at least with respect to acetaminophen and the carrots, that this isn't really a concern. I'm just curious, as you look at the whole spectrum of things that you've looked at, what gives you the most concern? Like which types of contaminants, which types of applications are actually the ones that you think are the ones we should be most focused on?
Yeah, so good question. So when we picked acetaminophen, we use it because it's the highest concentration all the time. And to answer your question, I think probably the antibiotics I think it might be concerned to people who are allergic to antibiotics at low levels. And there's probably a very few of those people who are allergic to this, like sulfonamides or penicillin. But again, there's really no study in human population, and because the first thinking is like it's so low. Yeah, so that's an area that might be done better by health people. Yeah, I'm not an expert in that.
Okay, are there other questions from the audience? Okay.
So on the ones where you were tracking the pharmaceuticals going into the soil, then ending up in the plant, we saw concentrations in the plant. I'm curious, what percent uptake does that represent?
Yeah.
I mean, is it a very small fraction?
Yeah, very small.
And most of it ends up in the soil?
Yeah.
And then is anybody looking at I guess the recalcitrants in the soil? Is it building up in the soil over time?
Mm-hmm. Yeah, I didn't show the other data. Yes, so we actually do also look at the soil depth profile, and we have lysimeters installed to see what's in the water fraction. I didn't put it there, but the percent that end up in the plant is below 1%, really small. Yeah.
Okay, well, thank you very much for a very interesting talk.